Recording now. So I asked you to kind of look at the origami you made, and not figure 4, I took that off. I thought you'd have your origami in front of you, but I forgot I collected it, so anyway. How is that similar to that of a ladder or a spiral staircase? That's what I asked you to do. See what you told me. Okay, DNA is similar because it's kind of a spiral, like a spiral staircase. It looks like a ladder a little bit. Twisted ladder, right? The way DNA spirals upward is how it looks like a spiral. So it looks like a stair or a ladder, but it looks like the ladder's been what? Twisted. Yep. So it's like a ladder, then it kind of goes up, but then it twists. So you don't have to climb it. You can walk up it like stairs, right? Okay. So this chapter is all about DNA, obviously. It's all about DNA, copying DNA onto RNA, wiggling through go out into to find to assemble, inform, to make proteins, peptide bonds to make proteins, right? I mess with you with that sometimes, I know. So what kind of RNA? We'll talk more about that later. We'll add a lot to that choral response. Don't you worry. mRNA, tRNA, and rRNA. Messenger RNA, transfer RNA, and I just dropped the tip of my doodler. I'm going to have to turn on the lights. Hold on. You know, about 10 years ago, I'd have been able to look down and see that. No, I'm just getting old. No, probably lacking in carotene. That's what it is. That's the protein. That's why you turn orange if you eat too many carrots. Carotene. What? Yeah, you can if it's got the yellow. If it's got a lot of carotene in it, you can. I haven't heard that, but definitely the carrots. You can't break down carotene, so you extrude it through. You extrude it. You get rid of it through your skin and your urine. When you sweat, you're actually peeing through your skin. So, yeah, there's urea in your sweat. Yeah, never thought of that, did you? So, let's get back on DNA. We got off on the urinary tract for a minute, folks. We're going to talk about what genetic material is made of. I know you all know it's DNA. We're going to talk about the experiments that led up to the discovery and what the role of DNA is, the shape of DNA, which you guys have already dealt with. And not today, probably tomorrow, we'll look at the information on DNA and how it's organized, and some of the other investigations that happened after they discovered DNA. So the structures for inherited traits are what are called genes. We know a gene is a small part of a large molecule called deoxyribonucleic acid. Everybody say deoxyribose. Remember we talked about the sugar the last couple days that DNA is made of? That is the name of the sugar. What does it end in? It ends in OSE. Fructose, sucrose, glucose, galactose. Those are all what? Sugars. Deoxyribose is a sugar. And it's the sugar that is part of a 3-molecule nucleotide, right? There's 3 parts to every nucleotide. So we have a sugar, a phosphate group, and a nitrogen base. The sugar in DNA is deoxyribose, so we call it deoxyribonucleic acid. Guess what the sugar in RNA is called? Just ribose, yeah. So it's called ribonucleic acid. So DNA is actually named after its sugar, so is RNA. Does that make sense? A little bit. ATP is another nucleotide. That's named after adenose, adenosine triphosphate. Okay? That's another sugar. So DNA is the primary material that causes inheritable characteristics in related groups of organisms. We understand it's how we transfer who we are as a species from one generation to the next. We've talked about mitosis. We've talked about meiosis. We've talked about fertilization with gametes and how we mix genomes together with chromosomes pairing up. We've talked about genetics and how you can inherit different traits or genes. Now we're talking about what those genes are actually made of. Okay? What makes them change from one generation to the next? What do we call that thing when something changes from one generation to the next? Evolution. Okay? And evolution is a very real thing. You can talk about evolution in many different ways. But evolution in itself, in its definition and what it is, can be observed in real time. We can observe evolution of viruses from one year to the next. We can observe evolutions of bacteria from one year to the next. Evolution only becomes controversial when you apply it to humans, which seems silly when I think about it as a scientist, but that's reality. That's the society we live in. Yes? Because I cannot, you got to kind of think about evolution in two ways. She asked, why is it called a theory? It's a theory because I cannot observe it with my real eyes. I can observe it in bacteria. So some forms of evolution I can watch happen in real time, and I can call them facts. Some, like human evolution, I have to rely on fossil evidence. I don't have a time machine. I don't have Mr. Peabody here, right? I can't jump back in the way back and go back and watch it happen. If I could, then I could call it a fact or a law, right? But because I cannot observe it with my own eyes in real time, we call it a theory. I will tell you, it is the one biological and scientific theory that has never been disproven by a single piece of scientific evidence ever to this date. So it's a very sound theory. We can watch it happen in all different organisms, but the second you do apply it to humans, we understand there is controversy there. So I don't even worry about that. Let's just talk about bacteria and how they change. You know what I mean? Or something else, starfish or whatever, you know. Martins. What? No, because then you get into multiverse and yeah. So kind of. A lot of our simpler ones, like the theory of evolution, is just accepted now. It's not really argued anymore. Not in the scientific community anymore. We'll say that. Okay. I do understand that there are non-scientists out there that don't believe in that. So, and that's a different issue. I don't even get into that in here because in the real world, outside of the classroom, yeah. Well, let me just tell you this. I'm required by the state of Michigan to teach evolution. If I wanted my job, I would still have to teach evolution whether I believed in it or not. So if I was in another state, that might not be the case. Here, we're educated in Michigan. We believe that we need to learn all of the theories of science. Okay? Which I'm happy, because I will tell you that evolution is what gives us a new vaccine for the flu virus every year, the fact that we can track the evolution of the viruses every year. So, folks, be careful just saying the general statement that evolution is hooey. Think about what you're saying. I don't care what you say about like human evolution. I really don't, because that doesn't affect us. But when you talk about looking at bacteria and viruses evolving, if we don't track those every year as a society and scientific community, we have a pandemic on our hands, literally. You know what I mean? So I always tell my students, be careful what you generally put a blanket over, because you could be covering up a cure for a disease. You know what I mean? So DNA is a very simple molecule. We'll get into evolution later. We don't need to have that discussion right now. That's like a chapter 17 thing. Okay? So there's three major experiments that led to the conclusion that DNA is the genetic material in cells. These experiments were performed by Griffith. We'll talk about him and his mice. Avery. We'll talk about how Avery supported Griffith's work. And then these two scientists that worked together, Hershey and Chase. You might want to just underline Hershey and Chase so you keep them together, because they are together. They had a partnership that they worked together on. So Griffith worked with two related strands of bacteria that cause pneumonia in mice. Pneumonia a bad thing generally? Yeah. You think how about this? What is pneumonia? You mean to tell me? Just fluid in your lungs. Can a lot of things cause fluid in your lungs? Bacteria, viruses, mold. Yeah. So be careful when you think about pneumonia as just being one certain bacteria. There could be thousands of different bacteria. Is there a problem back there? I'm saying pneumonia is caused by a lot of, not ammonia, pneumonia. Ammonia is the stuff you clean the house with. Your pee turns into ammonia if you let it break down the nitrogen cycle. But we're talking about pneumonia. That's a disease, a symptom of a disease. What? Yes. Remember the nitrogen cycle? You might have been absent for that. I think at the beginning of the year you missed some school. So, yeah, that's probably what happened. But that's probably what happened. I'm serious. That's probably what happened. We did cover this, though. So, anyway, looking at pneumonia, water in the lungs, it can be caused by a lot of different things. Sometimes it kills you, sometimes it does not. But here's what he found. He found that when he put one pneumonia in mice, it killed them. But then he discovered that when harmless live bacteria that normally didn't hurt you were mixed with the dead ones that he did kill that normally would kill you, they would then kill the mice. So let me explain that to you in kind of general terms. I got a chart in your notes that we'll talk about in a minute. But this actually led Griffith to call this thing that he saw and observed a transformation. And transformation, by definition, is a change in genotype. And if I change genotype, I can definitely change phenotype. And that's caused when cells take in foreign genetic material. Now, Griffith's experiments led to the conclusion that genetic material could be transferred between cells. Let me explain this to you in kind of just easy to understand terminology. So let's say I am working at the sewage treatment plant and I fall in to the fun stuff, okay? And I get out and I rinse off. Am I good enough to go now? No, I probably still have stuff on me. And then I say, oh, I'll fix it. I'll just come over here and I'll get some of this stuff. What's this? Hand sanitizer. I'm going to kill all that bad bacteria that could kill me. Let's say it's deadly bacteria, okay? All that bacteria is dead. Let's say I got all of it, 100% of it, okay? Shake my hand. So the bacteria on my hand is dead. He can't catch that bacteria, right? He can't, can he? So he can't. It's dead. Let's say you have bacteria on your hand, but it's perfectly harmless, but it's really hard to kill. Okay, so it won't hurt you at all, but you can't kill it. Shake my hand. So right now, something called conjugation is happening. I destroyed the cell membrane of all my bacteria. I killed it. What didn't I destroy? Everybody say it's DNA. It's DNA. Say conjugation. Conjugation is happening right now. My DNA is being absorbed by his good bacterial cells that normally wouldn't hurt them. And they're going to take in the stuff that normally would kill me, right? I could easily kill that bacteria, couldn't I? But now his good bacteria that don't hurt him are going to become super bacteria, right? That's called transformation. Does that make sense? Everybody say transformation. So they transform. It's like how a Decepticon turns in from a little toaster into something that'll kill you, right? Okay? So it reminds me of the Transformers. So you go through a transformation because one bacteria can take in the DNA of another, even if it's dead. Why can't our cells do that? I'm going to ask you this question. Why can't our cells go through conjugation and transformation just like that? Why doesn't it happen? Why do our cells not go through conjugation and transformation that easily? Think about what bacteria are and what we are. Bacteria are prokaryotes, right? Okay. Tell me why. Now, I don't ever want to alienate anybody when I talk about things changing and evolving, but we live in a changing and evolving world, right? We've gone from a world that's had dinosaurs to a world that's had mammoths, that's had large mammals, that now has small reptiles. if species did not have the ability to change genetically, we would all be extinct. Yes, sir. I don't worry about nonsense. I can't even. Yes, I know. There are some people that think that dinosaurs and people are running around together. I'm here to tell you there's literally 65 million years of rock between us and dinosaurs. No. I can't even have a discussion about that. Yes?
So, these cells that undergo transformation, do they reproduce in the same exact way? Do they create these cells just like the ones they transform into?
We're going to talk about that. So, it's a good question, though. The nucleus is missing in bacteria, right? So if there's no nucleus to protect the DNA, it's easily exchanged. We found that out, right, Mitch? Yeah. What? Yeah, the pilus. Yep, the pili. So bacteria actually have these little tubes. They shoot into each other, and then they stab in like Velcro, and then the DNA will flow between them, and it's called conjugation. They actually do this on purpose. It's how they evolve so fast. It's why bacteria will out-survive us way after humans are extinct. Bacteria were the first organisms on this planet. They'll be the last. They're that good at what they do. So the nucleus is what I was after. There's no nucleus in bacteria, right? So how does this whole thing happen? I have to correct one thing or it's going to drive me nuts. Just like a truck of pecans. And driving nuts, you know. Sorry. I gotta relabel that. It's going to drive me nuts. Okay. Now mitosis is the division of the nucleus. I stole this image off the internet earlier today because I wanted a better one than what I had. So take a look up here. Bacteria have an extra piece of DNA they carry around in them, with them, in them. Same thing. Everybody say plasmid. Anybody ever collect any cards, like baseball cards or anything like that? I collected baseball cards. Anybody ever do like the magic cards or whatever? Okay. Pikachu cards, Pokemon. Okay. Okay, I was trying to find something that more of us did. Isn't that Pokio? Pikachu? Pokemon? That's kind of sexy. Poke a woman. Pokachoo-choo. Star Wars cards, Clone Wars. I don't know. You guys didn't collect anything. What else? Webkins? No. What did you guys collect when you were little kids? Webkins? She didn't want to admit. She got 50 Webkins at home all lined up. What's their names? So, all right. So, you guys, let's say I got way off track there. Let's say that I'm collecting Pikachu cards, Pokemon cards. Okay, so I got like a set of 20, and Miss Lowe's got a set of 20, but we got different Poke people. What do we call them? Look at Ewald, she's out cold. Hey, good morning. Do you want breakfast? Oh, okay, all right. You sure? All right. Sure. Okay. I don't know. Anyway, so we're going to swap Pokemon cards. Thank you. Poke people cards. Okay. I know. Okay. Sneeze and chews. And I've got Sneeze and Chew, and you've got Pikachu. You got Bless You, and we're going to swap these, whatever the heck they are, cards. And so now I'm going to have a different deck of cards, right? So then I'll get, what do you use, like Dragon Ball Z power or what? All right. So we would swap powers, right? So then I'd be a little rabbit with a ball, and you'd be a monkey with a lightning bolt. I don't know how this works. So But this is essentially, you guys, what bacteria do all day long. They don't think about it. It happens, you guys, it literally happens as randomly as we sort our genes, which we don't sort them at all. They just do it randomly. And because of that, does that help them survive any environment? Yeah, because they don't choose, they don't select. It's random. They get all these different varieties and mixtures of who they are through transferring their DNA through plasmids. And you guys, because of that, they're able to survive and evolve all of these changing environments on Earth. There are bacteria that live in the sulfuric vents of volcanoes that it would burn our skin right off with the heat and the acid. So bacteria can live there because they've changed and evolved to do that. We actually are. There's more bacterial DNA in you right now and on you than is your own. That's creepy, right? There is actually more bacterial DNA in and on you because they're smaller than us, right? Think about how small they are. And they cover each little surface of us, yes. There's bacteria that live in our lower gut that break down milk for us. It's called E. coli. Okay? We are a living quorum of bacteria. Okay? So, don't believe me, don't brush your teeth for a day. You'll get a layer of what's called quorum sensing, all different sorts of bacteria forming a network and a community in your mouth. So, yeah. Now, let's talk about, I know, it makes you want to go brush your teeth, doesn't it? Yeah. So, now let me get on track a little bit here. Why is this important to us? Why should we care? Nothing we can do about it, right? Kind of. So we all know we should probably not use hand sanitizer. What should we use instead to wash our hands? Soap and water, because it does destroy the DNA. Okay? The antibacterial, and it rinses it down the sink too, but the antibacterial hand sanitizer, it's alcohol. It doesn't hurt the DNA at all. In fact, when you want to preserve alcohol or DNA, you put it in alcohol. It literally preserves the DNA. So hand sanitizer is probably not the best choice. The soap will break up the DNA. Now, let's talk about a problem that exists. Anybody know anybody with diabetes? Me too. Okay? So they got to have insulin, right? Where did insulin come from before we got the stuff that we make? Where? Yeah, okay. Yes, it comes from the pancreas. How about back in the early 1900s, we eventually got it from dead people. Let's talk about before then. What did they use? Pigs and cows, right? The only problem is a lot of people were allergic to those, and it killed them. And then your option then, if you were allergic to pig or cow insulin, you could get the cadaver insulin. So you take the pancreas out of a dead guy, squeeze it, right? And yeah, this sounds great. So, and then you'd inject that, and then it was like Russian roulette because eventually you get an infected sample, and goodbye, right? You get a bacterial infection in your blood. That's a bad deal. So now let's talk about what they do. Yep. There's beta islet cells. There's a few other cells there. No, because there's other cells there they need. Now, there's beta islet cells, all different cells there. Let's not get into that, though. Hold on one second. You guys, I do want to talk to you about how in biotechnology we use what bacteria do on their own to our advantage. What if I could take, you don't have insulin, or you don't have diabetes, right? Okay, so I'm going to take your gene for insulin because it's a good gene. Your body didn't kill it off earlier in your life. People that have type 1 diabetes, their body actually read those cells as foreign cells when they were younger. They made a mistake, and they killed them all. Their immune system destroyed all their cells that make insulin. They still have a pancreas. It just is missing what's called a beta islet cell that makes insulin. If I take her good gene, though, and I put it in a plasmid, everybody say plasmid, I have then genetically engineered a plasmid. And then if I put that plasmid into a bacteria, I have now genetically engineered one bacteria to carry and make the insulin gene. You get that? Remember, it's like my deck of Pikachu cards. Okay? Except this Pikachu card makes insulin. Okay? Now listen. Once it goes into this bacteria, bacteria are going to do what they do best, which is what? Reproduce and divide. And they divide through binary fission, and they literally make clones of themselves and everything in them, which means they make clones of your insulin gene, right? So in a biotech lab somewhere right now, today, so we don't have to inject dead cadaver insulin or cow or pig insulin into us, we have these bacteria that are being cultured that have been genetically engineered to carry the human insulin gene. And this bacteria will actually make, with its ribosomes, the protein insulin. Then we harvest that, purify it, and we sell it to diabetics so that they can inject it and they can break down their sugar. So now we get pure human insulin, and we don't have to worry about any of the other problems. So we kind of tricked nature's way of evolving into making our own selected gene. Is this cloning? Yeah. This is what's called gene cloning. I did this in here. Anybody see the Piglo, the jellyfish gene I had in here earlier in the year? What we do in biotech is we take an E. coli and we take a gene from a jellyfish and we put it in a plasmid, and then we put that plasmid in E. coli and we get it to glow in the dark under a UV light, just like the jellyfish does, but it's the E. coli that's doing it now, just to show kids how transformation works. So we actually get it to absorb and take it in, just like they do to make insulin. Okay? They've done this. They've made like kitties and puppies and pigs that glow in the dark with that gene too. So if you guys take biotech, we do that in that class. So is it a good thing that we study bacteria and the evolution of bacteria, the conjugation and transformation of bacteria? Yeah. Because if I were to have a diabetic child or a relative, I would want them to live, right? Who knows a diabetic right now? I got like 5 students. I don't want any of them dead, right? That are diabetic, yeah. I'm not going to say their names. That would be rude. Some of them don't want anybody to know. Allison Doak wouldn't care because she did her project. That's one. So if you're diabetic, this is a big deal, right? Even if you're not, you want your family members to live, right? No? So how do we figure out this is possible? Griffith observed it first. Did Griffith know about plasmids and DNA and all that back then? No. Let's talk about what he did know. He did know that if he injected this mouse with these R cells, the mouse was fine. This is a type of bacterium. Then if he injected another one with this nasty S cell, what did it do to the mouse? No more Mickey, right? But if he killed those S cells and then he injected them in, the mouse was fine. So normally the ones that killed the mouse, if he killed the cells, the mouse was fine. Mitch, remember I shook your hand? So if he mixed the ones he killed with the normally harmless ones and then injected the harmless ones, they weren't harmless anymore. They took on the attribute of the other harmful one, even though he killed it. He called this transformation. And he's famous for these experiments. He's in every biology textbook from middle school to high school. So he didn't know it was DNA. Avery, though, decided he was going to figure out what caused transformation. And that's what he's famous for. Avery proved what was causing Griffith's transformation to be DNA and not proteins or RNA. How did he do this? He basically isolated them. What he would do is he would destroy, let's say, the protein and the RNA and leave only DNA one time, and then the DNA and the RNA another time and leave only proteins, and then the protein and the DNA and leave only RNA in. And by process of elimination, we call this the scientific method with only one independent variable, right? He was able to determine it was DNA that caused transformation to occur. Yes, they isolate it. Yes, they do. They predict it, but they predict it based on the ones that they believe will proliferate that year by eliminating the ones that won't. So they were wrong this last year. I got the shot and I got the flu. So bad. A lot of people got the flu. No, that is a fallacy.
That's weird because I got the flu shot.
Yeah, there was a different one that came through that they didn't predict to be the one. Yeah. I did too. So many kids got the flu this year. Okay, so we'll talk about immunity for just a second here because this has come up. So listen, let me explain immunity to you. This is a primary response. This is a secondary response. The first time you see a bacteria, this is what will happen. The second time you see that same bacteria or virus, this is what will happen. Okay? This is, we'll say, the response intensity, okay? Response intensity. And we won't even give it a unit. Let's just talk about it generally. First time you see a virus, your body goes, oh, shoot, we better fight that off. It does that. And then it forms all these antibodies. And then when you fight it off and you get the viral load down, you don't worry about it anymore. Second time you see that flu or that virus, what happened? No. Quite the opposite. You already had the antibodies, you recognized it immediately, and you sent in the troops, full force. Okay? So your secondary response is way higher than your first response. This is basic immunity. Okay? Well, here's what I'm telling you. If you think you get the flu from the flu shot, you don't. What are you experiencing? The first response. You might get a little feverish, but you are not sick for like a month, right? Your body's responding. It's trying to heat it up. Your body doesn't know what that is. Your body recognizes proteins. And if your body was not recognizing surface proteins because they're still there in the dead one, right? Your first response you want to happen. You want to feel a little ucky when you get the flu shot. That means it's working. Okay? But if you're not throwing up, if you're not having diarrhea, if you're not in respiratory distress for 3 weeks, it worked. A lot of people say, oh, I got the flu. For how long? A day and a half. I felt like crap. Yeah, you didn't have the flu. You had the primary response. So now that you guys are all educated in that, when people tell you that, please tell them that they are wrong. And that meant the flu shot was working. Okay? It probably was not the flu that causes people to have, it's the respiratory flu that they try to predict every year. There are thousands of different types of flu viruses. Some of them are really bad. Some of them are mild. I got the flu this year. I did. It's bad. Oh, yeah, let's say. Yeah. You go through a week-long incubation period, then a 1 to 2 week I'm freaking sick period, and then a 1 week recovery period where you don't feel bad. So it's usually like a month-long episode when you get the flu. So Avery's experiments led to the conclusion that DNA is responsible for transformation in bacteria. So what do you have to know about Avery? He showed that it was DNA that was causing Griffith's transformations. Now, the other two scientists, Hershey and Chase, they found something neat out. They studied what are called bacteriophages. That word looks like bacteria, right? What if I told you phage means to eat? What do these viruses do? They eat bacteria, or they kill bacteria, right? So they're viruses that infect bacterial cells and cause the cells to produce viruses. Now, by using radioactive isotopes, Hershey and Chase showed that DNA, not proteins, is the genetic material in viruses. So here's what they did. If I could label sulfur and phosphorus, which of those two is found in DNA? Phosphorus. Remember yesterday the phosphate group on the side chain was black? Okay? So you find phosphorus in DNA. You do not find phosphorus in proteins. Then you find sulfur in proteins. Did I talk about sulfur in DNA? No. So there's no sulfur in DNA. Does that mean I can isolate proteins from DNA based on phosphorus and sulfur? So if I tag those two, I can track where DNA goes and where proteins go. Does everybody get that? All right. So what these guys did, this is called Hershey and Chase. This is a famous experiment. What they did is they allowed the bacteriophages that they tag those parts in to go ahead and infect cells. Then they took the cells and they tried to figure out what actually got into the cell to determine what was infecting the cell. And when they found that the proteins were only outside of the cell and the DNA had been injected into the cell, they found out that that's what the infectious component of a virus was. It was the DNA. Well, let me explain that to you in flu terms. We've all talked about having the flu, right? Everybody's had the flu at one time or another. No one's, if you lived in a population of people who have been to school for any amount of time, you probably had the flu. That's good. It means you got a good immune system. So, I'm drawing this terrible right now, but I'm drawing it. I'm going to actually go up to the board because my drawing's getting really bad. When you get the flu, here's what happens. Kind of gross and cool all at the same time. Let's do this. Let's do this. It's like a little alien landing on you. And then it actually has this thing that's called a probe. It's like a hollow tube. And on the top of it is this geometric head. This is a virus. What that virus will do, it has this DNA that it will squirt into your cell. And this is either DNA, and a lot of times it's RNA. One or the other. Okay? We copy DNA onto to wiggle out through us, go out into the, and find a, so let's say this is a ribosome. This ribosome is going to read that viral DNA and it's going to make what? Proteins. Are these proteins going to be ours? Whose proteins are they going to be? And then you're going to make more viral parts, literally. Divide this thing into 3 parts, kind of like minus the Z I just did.
Is that like what cancer does?
No. So if I make the head, the probe, and then the landing gear, proteins, and then inside of my cell they start to build up in all these different viroids, and then they come together, what's going to happen to my cell? It's going to expand. Let's talk about what happens when you get the flu. In your tummy, you have these folds like this. They're called microvilli. This is in your stomach and in your small intestine, like this. They increase surface area so you can absorb nutrients from your food, right? If your cells swell and you get sausage microvilli, what happens? Food has one option. It rots. Yeah. Imagine leaving food on your counter for 24 hours, then try like 4 days, then try a week. That's what's in your tummy. So you can't break down your dinner. It rots. And your body says, we can't squeeze rotted solid food through a small tube like my small intestine, so you do what? You chuck it out, and anything in the bottom end you get rid of because you can't deal with it. Okay? So you can't reabsorb water from your colon because your colon does that too. So your colon can't reabsorb water, so you get diarrhea, right? Good thing, drink more fluids. Yeah. They tell you what, bananas and crackers are the first things you're supposed to eat. So here's the thing, guys. If that's happening, your body's like, eject, right? Both ends. Says, we can't handle this. Yes? Because it's fluids, and you're dehydrated. Yeah. And there's nutrients in broth that you can absorb. Think about it. If you can't absorb and break down solid food, you probably can get a little bit of fluid and nutrients across the ends, right? Okay. So, no. So, you understand that? It's kind of gross to think about what's really happening inside of you when you get the flu. The viruses are nasty, right? Your body fights them off. So how do we get rid of them? We talked about the immune response, right? Okay, so if this starts happening, and I have we have things called B cells, and these will come by, and they will actually put a surface protein on here, okay? And we'll just, I can't draw, but we'll just say that's a skull and crossbones. That's a terrible one. All right, so this B cell, just put a tag on here. It's a death tag, right? It said, this cell's infected. What do you think our body's going to do to this whole cell? Yep, it's going to attack it. So then we have these other things called, they're actually called killer. Oh my gosh. They're actually called killer T cells. And a killer T cell will engulf this whole thing and digest it and break it down. Okay? You guys, this is happening in you right now millions of times. Okay? So how about this? I'm going to answer your question, I hope. Anybody ever heard of this guy? HIV, right? Bushmeat in Africa. So HIV is a virus that attacks who? Killer T cells. So if there's a virus that targets killer T cells, what can't we do? We don't have an immune system, right? We can't kill bacteria, we can't kill viruses. That's why it's called human immune deficiency virus, right? Because it breaks down your immune system. So usually people with HIV and AIDS do not die of that. They die of the actual, like the common cold or something simple. All right. So we've been looking at the shape of DNA. You guys made some origami. But a DNA molecule we know is shaped like a spiral staircase and is composed of two parallel substrands of linked subunits. Sorry. The spiral shape of DNA is known as a double helix. We'll find out in the next couple days that they thought it was a triple helix for a while. Thanks to Rosalind and Franklin, they figured out it wasn't. But each strand of DNA is made up of linked subunits called nucleotides. And remember, every nucleotide has three basic parts. It has a sugar, it has a phosphate group, and attached to the sugar there's the nitrogen base. Remember, a nitrogen base can be a G, a C, a T, or an A. All right, you guys. That's enough for one day, I think.